Hi everyone, my name is Bhavna Hurgobin. I'm a computational biologist based at Trobe University in Australia, and I'm also part of the ARC Medicinal Agriculture Hub. Today I will be talking about recent advances in Cannabis Sativa genomics research as part of the review paper that we published in 2021. So I will briefly talk about the importance of Cannabis Sativa research. I will also review existing cannabis genomics resources. In particular, I will talk about how we assess these resources, the findings that we made, and their implications for cannabis breeding. And lastly, I will touch on the application of plant genomics for cannabis breeding. So Cannabis sativa is a diploid species within the Cannabaceae family. It has a highly heterozygous genome comprising 9 autosomes and a pair of sex chromosomes. It is dioecious, meaning that it can exist as a male or a female plant. And broadly, it is classified as hemp or medicinal cannabis. An interesting feature of cannabis is the production of a wide range of secondary metabolites via the captate stalked glandular trichomes. Among these metabolites reside the cannabinoids and terpenes, which are the most sought after categories of secondary metabolites by breeders and pharmaceutical industries. And much of the success of the cannabis industry is actually attributed to these secondary metabolites. And with the relaxation regarding the legislation surrounding cannabis cultivation, there is now greater potential to accelerate cultivar development and export a wide range of secondary metabolites produced by this plant via the application of omics technologies. So one particular omics technology in this respect is genomics, which relies on the use of high throughput next generation sequencing data and some sort of a reference. And this reference can actually be in the form of a genome assembly. So at the time of publishing our paper, there were about 11 cannabis genome assemblies that had been generated and deposited into the public domain, but these are highly variable. For example, when we did this postcode analysis, which I'm showing the results here, we found that these assemblies had very different level of completeness, with CS10 CBDRX standing out as the most complete assembly. Having said that, some of the other assemblies that are listed here have also been key to confirming findings from earlier lower resolution studies, as well as uncovering important biology of the cannabis genus. And further improvement of these assemblies will assist in fully realizing their potential. We next set out to find out which of these publicly available genome assemblies contain key genes within the cannabinoid biosynthesis pathway, namely tetrahydrocannabinolic acid, or THCA synthase, cannabidiolic acid, or CBDA synthase, and cannabichromenic acid, or CBCA synthase, which produce the cannabinoids THC, CBD, and CBC respectively. In particular, we wanted to know how many copies of these genes were present in these various assemblies, given that substantial copy number variation has been reported with respect to these synthases. Firstly, we found that the number of synthase gene copies between genomes was highly variable, with some genomes having many more copies of the synthases compared to others. We also found that each assembly had at least one active or functional form of these synthases, and by active form, I mean the chemotype determining form of the gene, which are highlighted in the 3 different colored clades of this maximum likelihood phylogenetic tree. We also found that the active forms of these genes were highly conserved across cannabis genome. So for example, all the active CBDA synthases found within this clade were over 99% similar at the nucleotide level across the different assemblies. We then used expression data to assess whether copy number variation of cannabinoid synthesis plays a role in determining cannabinoid content. In other words, if one cannabis cultivar has more copies of THCA synthase, for example, compared to another cultivar, and if these copies are expressed, does it mean that the cultivar that has more copies produces more THC than the cultivar that has fewer copies? So our findings, which are supported by results from other research groups, seem to suggest that copy number variation may not actually affect cannabinoid content. So in our analysis, we aligned short-read RNA-seq data from 9 medicinal cannabis cultivars generated by Zager et al. From a study back in 2019 against the CSN reference genome, and we assessed the expression levels of the cannabinoid synthesis in these cultivars. So as you can see, based on the cannabinoid content, the majority of these cultivars produce high quantities of THCA, with the exception of one cultivar, which is called Canat2, which predominantly produces CBDA. And if we look at this heat map of gene expression, we can see that one CBDA synthase in particular, so the one that's in this green box right here, has much higher expression compared to the other CBDA synthase gene copies. And this seems to be more pronounced for Canat2, which has a higher CBDA content relative to the other cultivars. So this CBDA synthase copy is actually the active form of the gene, and therefore it appears that we only need this particular gene or copy of this gene to be present and expressed for CBDA synthesis to occur. Another interesting finding was the expression of the CBCA synthase genes across the 9 cultivars, in particular, the higher expression of the 2 genes that we have highlighted here with the black arrows. However, the presence of cannabichromine or CBC, which is synthesized by CBCA synthase, was not detected in these cultivars. Instead, it was the presence of THCA that was observed. So what we think happened is that the short reads that belong to the active form of THCA synthase in these high THCA cultivars were forced to map to the CBCA synthase loci in the CSN reference genome. We think this happened due to 2 main reasons. Firstly, the CSN reference does not actually contain the active THCA synthase locus. Secondly, CBCA synthase and the active form of THCA synthase are highly similar, sharing over 96% nucleotide identity. So to investigate this, we aligned the reads from the high THCA cultivars against 2 references. The first one was the CSN reference, and the second one was the maternal Jamaican lime reference, which is derived from a mixed chemotype that produces both CBD and THC, and therefore contains the active forms of CBDA synthase and THCA synthase. And for simplicity today, we are only showing the results for one of the cultivars, which was white cookie in this case. So the top panel represents or shows the alignments against the 2 CBCA synthase loci that were the most highly expressed in white cookie. And the bottom panel shows the alignments against the functional THCA synthase locus. So we can see that although the reads technically align to all 3 loci, the alignments at the 2 CBCA synthase loci contain many single nucleotide polymorphisms, which are shown here by the vertical colored lines. And we can also see that the read coverage of these 2 loci is not as even as what we see against the THCA synthase locus. So the results from this analysis point to 2 main things. Firstly, the use of short read methods for genomic analysis, as was the case here, can create misleading artifacts because short reads that originate from highly similar regions of the genome frequently cannot be distinguished. And secondly, the choice of the genome assembly can also impact our interpretation of results. So the second point actually brings us to the concept of the pangenome. So numerous studies have shown that there is a considerable amount of structural variation among individuals of the same species, which are manifested in various forms that are shown here. And we saw earlier that the cannabinoid synthases tend to display presence, absence behavior, and this could also be the case for other agronomically important genes in the cannabis genome. So this suggests that working with a single reference at a time can limit biological interpretation. And this is where the pangenome becomes handy because it represents the full repertoire of genes in a species. So it consists of the core genes, which are present in all individuals. And then we also have these variable genes or dispensable genes, which are present in some, but not all individuals. So an example of a pangenome for cannabis would basically be a single reference that contains a non-redundant set of core genes that are present across cultivars, as well as all the other genes that show presence, absence, variation in the species. So this pangenome would increase our understanding of the extent of gene content variation in cannabis, as well as forming the basis for cannabis breeding programs. For instance, the inclusion of diverse genotypes in wild cannabis populations would facilitate the identification of early marker genes in the variable genome of cannabis and drive the process of heterosis to create resilient and high-yielding cultivars. Additionally, combining gene presence, absence, variation, and pangenome white slips would lead to a more accurate identification of causal variants associated with traits of interest. So to summarize, what we saw today was that several genome assemblies are available for Cannabis sativa, but they are variable in terms of their quality. Extensive copy number variation of cannabinoid synthesis is present in cannabis, but the association between copy number variation of these syntheses and cannabinoid content may not be significant. And the choice of the assembly for genomics analysis can impact the biological interpretation of our results. And pangenomics has great potential for accelerated breeding of cannabis. And if you'd like to read more about our research, you can find our paper at this DOI. So to finish off, I would like to thank all the present members of the Lucy Lab, as well as the members of the ARC Medicinal Agriculture Hub and all of our industry partners as well. Thank you for your attention. If you have any questions, I'd be happy to answer them now.